Welcome to you from ChemHelp ASAP. Molecular biologists not only identify proteins that may serve as drug targets, molecular biologists also develop tests that determine how well a molecule binds to a protein. These tests are called binding assays. Binding assays measure the strength of the interaction between a protein and the drug candidate. This is also called the potency of a drug. A drug that binds its target more strongly is said to be more potent or to have a higher potency. We will not define these terms, but IC50, KD, and EC50, among others, are terms that are associated with numerical values and different ways to express drug potency. The term potency can be confusing. A potent molecule does not necessarily have a strong effect. These are not equal. Instead, potent molecules are able to exert their effect, whether strong or weak, at low concentrations. Active at low concentrations. So, efficacy and potency are not interchangeable terms. The assay developed by the molecular biologists will be used throughout the drug development process. All new molecules will be tested in the assay to determine whether the new molecules are more or less potent than previously studied molecules. Although potency is not the only factor to consider, molecules with higher potency are preferred over less potent molecules. A molecule with higher potency requires a lower concentration in the body to exert its effect. A lower concentration means the patient can take a smaller dose. A lower dose normally means fewer side effects, or more properly, fewer adverse drug reactions. Therefore, a more potent drug is normally a safer drug. One of the approval criteria of the FDA is safety. Let's return to the idea that efficacy and potency are not equivalent. When the molecular biology group identifies a drug target, the group believes that the target protein can control the disease in the patient. Unfortunately, predicting the action of proteins is not always easy, and an organism might have alternate proteins for the same job. The process of confirming the value of a target for treating a disease is called target validation. A target cannot be truly validated until phase 2 clinical trials. It is only in phase 2 trials that the development team will confirm that binding the target with a molecule leads to a therapeutic effect in humans. In other words, potency can be measured in a lab. Efficacy requires human testing, phase 2 trials. The drug target binding assay is a vital tool for a successful drug development program. The assay allows the chemistry team to know which molecules have the highest potency.